The apparatus use. What we call this apparatus? So we call this as electrodes. Then the next part, because the common term for anode and cathode is electrode. If it is specifically at a positive one, positive plate, then we call anode. If it is specifically on negative, we call cathode. But collectively, common term is electrode for both anode and cathode. On the diagram, sketch how a sample of a gas on a diagram, sketch how a sample of one of the gases could be collected. So how we can collect, and yeah, we will use an inverted measuring cylinder, but not measuring cylinder or even a gas syringe. Normally when we use gas syringe is used when we have a container, a sealed container, and a gas is given off, or in that case we are using a gas syringe. But because here it is difficult to attach a gas syringe, that's why we don't attach a gas syringe. We can attach a gas jar, or we can attach a measuring cylinder which is also having some liquid inside. Because how we collect the gas in this technique, like this is a measuring cylinder, we completely fill this measuring cylinder with the electrolyte, and then what we do, we invert this measuring cylinder on the electrode. So when we invert this measuring cylinder on the electrode, some of the electrolyte which we filled in the measuring cylinder will go out as the gas take that place. This is how we collect the gas given off, not by a gas syringe, because it is difficult to attach a gas syringe here. Because practically I cannot connect a gas syringe, that will be not correct. Give a test for oxygen. How we can test the presence of oxygen? So we will place a glowing splint near the oxygen, and the result will be, the test is glowing splint which will relight or rekindle. So in short you can write relight. The gas collected at a positive side turned lime water. The gas collected at the positive sides turned lime water milky. Based on this observation, what gas was present? If the gas is turning the lime water milky, so that gives an idea that the carbon dioxide gas is there, because the carbon dioxide is the only gas which can turn lime water milky. Then suggest how this gas was formed. Now the question is how carbon dioxide is formed. In the question already in the beginning, they already mentioned the two gases which are given off are hydrogen and oxygen. But you can see here,
Oxygen react with the electron.
yeah,
With the
oxygen,
carbon and the electron.
yeah, that's right. Basically what happened, the oxygen gas which is formed at cathode might react with the carbon, and that result in a formation of carbon dioxide. So oxygen formed reacted with the carbon electrode, result in a formation of, because the oxygen gas, as this is a dilute sulfuric acid, so ions which it contain hydrogen ion, hydroxide ion, H ion, and SO4 ion. So hydroxide ion, negative ion, will move towards anode, and when it will move towards anode, it will produce oxygen gas, and this oxygen might reacted with carbon, result in a formation of carbon dioxide. So O2, the oxygen reacted with the carbon anode. The next part, a solution of a dilute sulfuric acid was electrolyzed for 1 hour. Like we did electrolysis for 1 hour, suggests why the pH of the solution decreases during the electrolysis. What could be a reason why the solution pH decreases?
Because the hydrogen ions increased.
The pH decrease means it become more acidic. Why it become more acidic? If you check the electrolysis, when we are doing electrolysis of sulfuric acid, look what happened. It contain hydrogen ion, sulfate ion, and hydrogen ion and OH ion. So what is happening? H is being removed, it will move towards the cathode. OH is being removed, it will move towards anode. So what it become, because if H and OH are being removed, it means it will become more concentrated. As the water is H and OH removed, means the water is removed, so it will become more concentrated, and more, concentrated acids have lower pH than dilute acids, or concentrated acid solutions are more acidic than dilute acidic solutions. So the reason why it turned pH decreases, or why it turned more acidic, because water is removed, so it changes to concentrated sulfuric acid, and concentrated sulfuric acid will have a lower pH as compared to dilute sulfuric acid. So this was question 1, and question 2, a student investigated the reaction between the dilute hydrochloric acid and the two of the aqueous solution of sodium hydroxide, so which are labeled O and P. So two solutions of sodium hydroxide, we don't know their concentration. One is O, another one is P. And then, yeah, neutralization reaction is there. Two experiment was carried out. Experiment 1, what we did, in experiment 1, a burette was filled with a dilute hydrochloric acid. So we fill the burette with hydrochloric acid. Hydrochloric acid, we know its concentration. So in the burette we have HCl or hydrochloric acid. And in a conical flask, we'll take the sample of sodium hydroxide. And because acids and alkalis are colorless, to know when all the acid completely neutralize the alkali, we use or add an indicator. The technique is called titration. The reaction between acid and base is neutralization, and this technique is known as titration. So a burette was filled with hydrochloric acid. The initial burette reading was recorded using a measuring cylinder, 20 cm cube of, so we put 20 cm cube of sodium hydroxide, and we pour HCl from the burette until indicators show a color change, and we, while swirling the flask, to make sure that they thoroughly mix with each other. Now the question is, using the burette diagram, record the reading in the table and complete the table. What is the initial burette reading, starting burette reading? We read the lower meniscus, that's 4.1. What is the final burette reading? There are many types of indicator. It is not necessary like you know all the type of indicator. You should understand the fact that indicators are the substances which show a color change when they are in different medium. That is the important thing which you should know. It's not like there are many indicators that sometimes use their name, so you don't have to bother that you took or you studied this indicator, because not all of the indicator you are studying. You're just studying an idea that what is the concept of indicator. What is the difference? 29.6 and 4.1. What is the difference? So 25.5. Then a conical flask was emptied, rinsed with the distilled water, and experiment 1 was repeated using P instead of O. So we did the same experiment, but this time we repeated. with O, so this was 29.6, this was 4.1, and we were getting 25.5. The initial burette reading, what is the initial burette reading for experiment 2? 24. What is the final burette reading? 29.1. And what is the difference? Initial was there at the bottom, so 24, 29.1. And when I take the difference, it will be 5.1. Now, look, if you have a conical flask, okay, and you have unknown solution, in the beginning we took solution P, solution O of sodium hydroxide, and then the 20 cm cube of solution P, in the conical flask. And how much HCl is required for the first reaction? You need 25.5 cm cube of HCl for a complete reaction. For the second reaction with P, you need only 5.1 cm cube of HCl for a complete reaction. Which solution is more concentrated? Sodium hydroxide solution, solution O or solution P? Which solution of sodium hydroxide is more concentrated? Which solution contain more particles or more concentration, O or P, for sodium hydroxide? The correct answer is O. Why? Because you need greater amount of HCl for a complete reaction, so it means this has more particles, that's why you need a greater amount of HCl for a complete reaction. P is having a smaller number of the particle, that's why a smaller amount of HCl is needed for a complete reaction. Because when I add 5.1 cm cube of HCl, it shows a color change and it neutralizes by only 5.1. But for O, for a complete reaction, I need 25.5 cm cube of HCl for a complete reaction with O. So what it shows that O is more concentrated, or O contain more particles as compared to that of P. Is it clear?
Sir, doesn't more particles means less use of hydrochloric acid?
It's not about yield. They are not asking about product. The question, because there will be a question in which they ask which solution is more concentrated. So how you work out which solution is more concentrated? The unknown solution. You have unknown solution O, which is 20 cm cube, and you have unknown solution P, which is also 20 cm cube. For a complete reaction, for completely neutralize O, I need 25.5 cm cube of HCl for a complete neutralization. But here, for 20 cm cube of P, I only need 5.1 cm cube of HCl for complete neutralization. So which solution of sodium hydroxide, solution O or solution P, is having more particles? So it means solution O is having more particles. That's why it needs greater amount of HCl for a complete reaction. Yeah, concentration of HCl is same for both. We took the same HCl in both reactions. The only thing which we change is what is there in a conical flask. Now, what type of chemical reaction occur when dilute hydrochloric acid react with sodium hydroxide? What we call this reaction when acid react with base?
Neutralize.
We call that as neutralization. SATIUN or ZATIUN, both are correct. You can write. which solution of sodium hydroxide is more concentrated, and explain your answer. So which solution is more concentrated? Solution O is more concentrated because it need greater amount of HCl for complete reaction. So answer 1 mark is for just writing O, like identifying, and second mark is for reason, because it require for complete reaction or completely react. Then the next part, how many times more concentrated is the solution of sodium hydroxide than the other one? So it's about 4, because what happened, this was 25.5, the difference, and this was 5.1. So when I compare the ratio, so that will be 5 times, because 25.5 divided by 5.1, so it is 5 times higher concentration or more concentration. Now, if experiment 2 was repeated using 10 cm cube of solution P, what volume of hydrochloric acid is needed? This was done when you use 20 cm cube, because we use in experiment 1 we use 20 cm cube of O, then experiment 2 we repeated with P and the same amount 20 cm cube. So what they're saying, example, in a conical flask, when I have 20 cm cube of P, I need 5.1 cm cube of HCl. Okay. Now they're saying if I repeat this experiment with 10 cm cube of P, like half the volume, so how much I need, I can use a ratio, simple ratio I can use. So 2.55, the simple ratio will be there, but because for burette, the thing is, burette can, smallest value which the burette can record is 0.1 cm cube. So even using a ratio, like example, if 20 cm cube of P, I need 5.1 cm cube of HCl. So 10 cm cube, I need X cross multiply, answer will be 2.55. But I cannot write 2.55 cm cube because burette maximum, the smallest value which we can measure is to 1 decimal place. So you can write 2.5, that is acceptable, or you, if you round it off to 2.6, that is also acceptable. So either 2.5 or 2.6. Normally when we rounding off rule is there, when there is a 5 and we have a odd number next to that, so we add 1, and if there is 5 and even number is there, we do not change. That's the rule of rounding off, but even 2.5 they accept. The next part, what would be the effect, if any, on the volume of dilute hydrochloric acid in experiment 1 if a solution of a sodium hydroxide were warmed before adding to hydrochloric acid? Is there any amount or effect of temperature, amount of acid needed? So effect, there will be no effect. Reason for that, it can affect the rate, but it will not affect, because we have the same number of the particle. Temperature will only affect the rate, but it will not affect the amount of reactant required for a complete reaction. So effect on volume, there is no effect. So temperature affects the rate only, not the amount of reactant particle. So you need the same amount of acid for a complete reaction, even you carry out reaction at high temperature or low temperature. What would be more accurate method of measuring the volume of sodium hydroxide? We use a measuring cylinder. What we should do? So we should use a burette. Pipette, normally pipette is like 25 cm cube pipette is there normally in the lab which we use. So burette is the one we can have like zero point 0.1 cm cube. So we can use a burette which is more accurate. Suggest how the reliability of result could be checked. How we can check our results are reliable, our results are accurate. Yet repeating the experiment and taking an average. So repeat the experiment and take an average, increase the reliability of the result. Now, aqueous sodium hydroxide react with aqueous sodium hydroxide react with calcium chloride to form a white precipitate of calcium hydroxide. Use this information to suggest a different method to find out which solution of sodium hydroxide is more concentrated. Use this information to suggest, like how we can use this information and work out which solution is more concentrated. So they mention when sodium hydroxide is added to calcium chloride, it will form a white precipitate. So how we can use this technique or how we can use this method to show that, no, using this technique, not about electrolysis, they're asking using this idea. So what we will do, we'll take equal volume of the two solutions, add calcium chloride, so take equal volume of solution O and P. Then what we will do, we will add excess of, how fast can be done, but precipitate formation is practically difficult thing to work out the rate of formation. You can also use a technique, rate of reaction, add excess of, or you can add even excess or take equal amount or add equal amount. Then measure the mass or thickness or height of PPT. The experiment, the solution which form greater amount of precipitate is more concentrated. So the solution which is having greater amount of precipitate or greater mass of precipitate, that is having a greater concentration. Is it clear, this simple experiment? Okay, the next question, it's about identification of the ions. Uh, Two solids Q and R, which were both salts, were analyzed. Q is a zinc bromide, and test on Q are given. Zinc bromide salt is there. So ZnBr, zinc 2 bromide, ZnBr2. Solid Q was dissolved in a distilled water. The solution was divided into 3 equal portions and tests were carried out. We have a zinc salt and we add sodium hydroxide, drops of sodium hydroxide. What will be the observation if we add a small amount of sodium hydroxide to a zinc salt? So it will give white PPT. that is result in a white precipitate. Excess of aqueous sodium hydroxide was added, like more than enough sodium hydroxide is added. What will be the observation? So precipitate dissolve to form a colorless solution. When white precipitate dissolve, it will form a colorless. If colored precipitate dissolve, then it will form a colored solution. So dissolve to form a colorless solution. Then we add an aqueous ammonia. If we add aqueous ammonia to a zinc salt, what is the observation? So it will form white PPT. Again, in exam you can write PPT instead of precipitate. And if we add an excess more than enough, what is the observation? The precipitate will dissolve and it will form a colorless solution. Part C, a dilute nitric acid followed by a silver nitrate. This is a test for halide, chloride, bromide, iodide. What is the observation if bromide ion is there? With chloride it give white, with iodide it give yellow. So with bromide it gives cream precipitate. 1 mark is for mentioning the color cream, and the second mark is for mentioning a PPT or a precipitate. The term precipitate is used for a solid form when two solutions are mixed together. Tests on solid R. Tests were carried out and the following observations were made. A flame test about R, they did not give any information, so we have to work out what is R. So the first one, this one is a test for halide ion. Dilute nitric acid and silver nitrate is a test for, because if you have silver nitrate, when you have silver nitrate and if there was a chloride ion, so silver will react with chloride ion and it will form silver chloride, that is white in color, white PPT. But if you have bromide ion and you add silver nitrate, AgNO3, this will result in a formation of silver bromide, AgBr, which is cream color precipitate. And if you have iodide ion, you add silver nitrate, AgNO3, silver nitrate or lead nitrate, it will give silver iodide, which is yellow PPT. So you can repeat this test with silver nitrate or lead nitrate. Test on solid R. Flame color is yellow. So if it's a flame color yellow, this gives an idea it's sodium. R was dissolved in a distilled water. The solution was divided. Dilute nitric acid followed by barium nitrate. So this is a test for sulfate ion and it does not give any change. So what we conclude, you can say there is no sulfate ion. Dilute nitric acid and aqueous silver nitrate. Again, this is a test for halide. So yellow precipitate, it means it forms silver iodide. So the that is, it shows confirm that there is a iodide ion. So it's of 2 marks. So identify solid R. It is sodium iodide. Then this was the last part. This is the last question. Yeah. When barium hydroxide is added to ammonium chloride, a reaction takes place. Describe an experiment to show that this reaction is endothermic. So if we want to show an endothermic reaction is there, when a reaction there is a temperature change, so endothermic, so the surrounding temperature will decrease. Surrounding here refers to temperature of the mixture. So we need a thermometer for this. And we have a solid sodium barium hydroxide is added to solid and a reaction take place. So first what we have to do, we have to take equal amount of barium hydroxide and ammonium chloride. Measure the initial temperature of each. Mix in a test tube and record the final temperature. And if the temperature, because if it's an endothermic reaction, it will absorb energy. If the temperature decrease or the test tube feel colder, it is endothermic. How could you show whether or not the final mixture contain ammonium ion? So the test for ammonium ion, add sodium hydroxide and heat. A pungent gas is given of irritating gas, which will turn damp red litmus blue. Uh, what's your question if we do this experiment if reaction is taken in a water bath?
Sir, in some questions like MCQs, they
Hmm.
ask in exothermic or endothermic
Yeah,
reaction, what is the temperature of the water? Is it colder or hotter, or
yeah.
in
So
that sense?
in that sense, like example, what happened if you have a mixture of barium hydroxide, say aqueous solution of barium hydroxide and ammonium chloride. When you mix them, initially if the temperature is 20, if the reaction is endothermic, the final temperature will be less. Why? Because the water, you are getting the temperature of the water molecule which is surrounding. The reactant molecule absorb energy from them and they undergo a reaction. So temperature will decrease if it's an endothermic reaction. Like some of the sweets or mouth freshener, when you are eating those sweets or mouth freshener, in your mouth you will feel cooling effect, colder. Basically, that is also an endothermic reaction, because what happened, that sweet or mouth freshener is absorbing or taking energy from your mouth, so your mouth will feel colder. A chemical reaction take place, and the particle take that energy, and your body loses that energy, so it will feel colder. Same thing like happen in a mint sweets or tablets. Is it clear?
Okay, sir. Thank you very much, sir. Like, if the question is asked, like, can I write in this sense, like in a water bath?
Yes, you can write that in that.
Okay, sir.
Because
Thank
in
you,
the
sir.
question, yeah, what we measure for initial temperature, we measure the initial temperature of the two solids. Like what is the temperature of initial temperature of sodium hydroxide and the initial temperature of ammonium chloride. We measure their initial temperature, we mix them, and because these are solids, so we will stir or mix them, or using a mortar or pestle, we can even crush them for having a large surface area and observe or measure a temperature change. So this was paper 6 of February March 2017, variant 2.